All right, guys, welcome to AP Biology and the first lecture. And you should have in front of you the note packet, and you should be taking notes. Anything I write, I expect you to write. Anytime I'm talking too fast and you're not able to listen, remember you can always hit pause, you can rewind. That's the great thing about having these lectures taped. And remember, if you want to write additional information to help you understand something a little bit better, then feel free to do that. All right, bear with me. I'm going to be using a different recording method than what I'm used to record this lecture. And I'll do my best to try to write as neat as possible. We're going to start out with something that you guys are familiar with, inquiry, because you're going to see that we're going to do a lot of experiments throughout the year. And a lot of your questions that you have on the exam are based on research. Now, anytime we're doing research, we have to have an independent variable. So to help you remember what the independent variable is, I want you to think of I for independent and I for what I changed or I made different when I set up the different parts of the experiment. For the dependent variable, I want you to think of D for dependent variable. Okay, so D for dependent variable, D for data. So the data that you collect at the end of the experiment and then analyze, those numbers will represent what the dependent variable is. Now remember, your dependent variable, it needs to be data. It needs to be a number. So it has to be something that's measurable by a number or quantifiable. That means that you can get a number as your dependent variable. You need to be able to list me constants that need to be kept the same or discuss variables that need to be held the same is what we're going to write there. Okay, for each of the experimental groups. The control group gives us different information. Sometimes the control group gives us data that we can use to compare to the other experimental groups to see if those changes that we're making are affecting the data that we're collecting, the dependent variable. Sometimes we use the control group to help us identify if there's other factors that are changing. So if some of our constants are changing, sometimes our control group can indicate that. So that means that you can't trust the data that you're collecting from your experimental groups because you have these outside factors affecting the data. And then sometimes it helps us determine if our equipment and our materials and maybe the methods or design of our experiment is going to collect data as we expect. Now, there's two types of control groups, so this is going to be new from your honors class. We have the positive control group and we have a negative control group. So let's go ahead and just write down, and then we'll look at some examples of positive and negative control groups, right? So a positive control group is going to be one part of your experiment. And when you look at what happens in that part of your experiment, you will see a change. Something is going to happen. Okay, so you will get a result. You'll get a change as expected. Now, if you don't get that change like you expected, then again, that's going to be telling you one of these things. Actually, more like the last two down here. It's going to be telling you, okay, maybe there's an outside factor, or maybe some of the material you're using, like the buffer solution or the dye that you're using, is outdated and it's not working like it should. The negative control is the setup of your experiment, and you're going to see no change happen in that part of your experiment as you expect. If you do see a change, again, that's going to mean like these possibilities right here. There's an outside factor that is affecting your results, and you cannot trust the data that you're collecting from your experimental groups. All right, so let's go down to this first essay question. And this has to do with, what we're talking about here. Inquiry, IV, DV, control group. So take a second to pause and get familiar with this experiment and see if you can figure out what is the IV and what is the DV. Okay, so in a table, a quick way to figure out what is the IV is the IV always comes before the DV. So it looks like the type of fluid that we're ingesting here, okay? Or I am varying the amount of alcohol. So that's what I make different between the different groups of my rats that I have set up. So you would say that the type of fluid ingested is the IV. You could also say the amount of ethyl alcohol is being varied. That could be an example of your independent variable. The dependent variable is always the data that you're going to collect. So D for dependent variable, D for data. So if you think about it, so here's my data. this 3.5, the 3.8, and the 4.7. So remember that our DV is always going to be represented by a number. And it's what we're going to record and put down in our table, either like during the experiment as it's ongoing or at the end of the experiment. So these numbers wouldn't be here yet unless we've done the complete experiment. So they represent the dependent variable. Okay, now going through this first essay question, a few things that I want to point out. Notice that we have bold words, and the bold words are going to help you determine how to get your points. So for A, all I need you to do is pose or write a scientific question that's being investigated, state a hypothesis, describe the effect, and justify. So this is a 4-point question. Notice that whenever I write my answers, I write them with the same letters that we see here, A, B, C, and D in the question. So then a grader knows exactly where to go to give me a point for A, for B, and so on. Okay. Now, when you go to write a question, you need to make sure it contains the IV and the DV. So that's why we had to figure that out first when we looked at this experiment. So how does alcohol consumption, right? So there's my IV, affect urine output, which is the DV. And you maybe have been taught that to write a hypothesis, you need to use an if and then statement, but that's not correct, or that's not necessary. All you need to do is talk about what is being varied. So, what is the IV? And then give me a prediction about what's going to happen to the DV. So, I predict that an increase in alcohol consumption increases urine output. So, again, we're talking about here is the IV, and we've indicated that in this case, we're increasing alcohol consumption, and then we're predicting about the DV. All right, C says, describe the effect of ethyl alcohol on urine production. So this is an easy answer, and I just wanted to put this in here to show you how short of an answer you can write down. I know this is an essay question, but that doesn't mean you have to write paragraphs to get your points. So a good answer to write would be an increase in alcohol consumption. So here's my IV, increases the urine production. I got that from the data that we see up here. And again, I've written a simple statement. It is a sentence, and I don't have anything extra in it. And then moving down to this last part, it says the control or justify the use of water as a positive control. Okay. So I get for answers on this question, students will say, well, water is the control group because it is the norm. And that's what they want to use as their justification. So it is the normal thing that rats will drink. So therefore, that is our control group. But you're not going to get any points for that answer. Our answers are going to have to look a little bit more and related to what I have indicated up here, these things right here. So is the control group giving me this information, this information, this information? And you have to be specific to the experiment. So here's what I mean by that. So if we go down and we look at the answer here, it states that the control establishes my normal urine output when consuming water. Okay, so we are making this very specific to the information that is in this experiment. So the control group, which would be the situation where we're giving water to the rats, is establishing what normal urine output is. So we can use this data to compare to the other ones to see if adding alcohol or changing alcohol consumption will affect the urine output. Okay. You can see that my answer here that I wrote here in D is basically doing what we have up here in this very, very first one. Okay, so right here, this is kind of what I stated, but I was very specific to the experiment. So the control group gave me data so that I can see if adding alcohol is having a significant effect on urine output compared to water. Now going to the bottom here, I'm just giving you some tips that I already kind of talked about here while answering this essay question. Again, note the bold words. That's how you can determine how to get your points. Now this isn't a 3-point question, it's actually a 4-point question. Notice that I've sectioned out my answers, A, B, C, and D, just like the question. We don't want intro statements. We don't want conclusion statements. We want you to get to the point. So if it says, write a question, then your answer is going to be a question is, and we want it short, but we want it to the point. You have a limited time to answer essay questions, so we need to make sure that you're being very efficient whenever you're writing your answers to your essays. Also, note every single answer that I have there, A, B, C, and D, they're all complete statements. If you just give a word, or if you draw a picture, or you make a list or bullet points and they're not complete statements, then you're going to get 0 points. For example, let's say there was an E and it said identify the dependent variable. So in your answer, if you just said E and then you wrote urine output, you would get 0 points. You have to indicate that the dependent variable would be urine output. Remember, you don't have to use if and then. All you need to do is talk about how the IV is changing or being changed and then predict about the DV. All right, moving on. You've probably been taught that when you do an experiment, you have one independent variable. So you're changing one variable between the different parts of your experiment. But however, in research, to save time and save money and resources, sometimes we can run multiple independent variables at a time. So on the AP test, they like to give you examples of experiments or research and where they're testing more than one independent variable at a time. So to give you an example here, it looks like, if we're going to kind of get ourselves familiar, it says that we're going to measure the effect that light has on photosynthetic rate, and we're going to use a blue dye called DPIP. And it's going to start out blue, so I'm just going to indicate DPIP is a blue dye, and it's going to turn clear if photosynthesis is happening in the test tube. So here's kind of the setup of the experiment. I'm just going to draw it over here to the side a little bit so you understand what they're putting in their test tubes. So here's the test tube, and they are putting chloroplast in these test tubes. We're going to do some experiments similar to this, and they're putting that blue dye in there with some water. So initially this test tube is going to look blue. They're going to expose it to light, and over time we should see that, so let's indicate over time, we should see that our test tube should end up looking clear. We still have those green chloroplasts in there, okay? So if photosynthesis is happening or being performed by the chloroplast, our test tube, that solution in it is now going to look clear. Okay, so if you set up these test tubes, let's take a look at 1 and 2. Notice that they've done something different. So in test tube 1, they're using just normal chloroplast. And in test tube 2, they've taken those chloroplasts and they've boiled them. So they exposed them to high temperatures. So they've changed one thing between those two test tubes. So if I just look at the results between test tube 1 and 2, I have an experiment. They want to see how boiling the chloroplast affects the rate of photosynthesis. So I have an IV and I have a DV, okay? Now, if we take a look at these questions here, let's see some of the other independent variables that are being tested here. It says, identify the test tubes that you would compare to determine if chloroplasts are necessary for photosynthesis. Well, we need to set this up without chloroplasts. We're going to put everything in there. So we need to put the DPIP, water. We need to expose it to light because we know light is necessary for photosynthesis, but without chloroplasts. And notice that is the test tube number 5. Okay. And now we need to compare it to our test tube number 1. Same setup, but we have chloroplast in there, okay? So we would write our answer, and again, we're going to write a complete statement. To determine if chloroplasts are necessary for photosynthesis, we would compare test tubes 1 and 5. If you just listed A and then you wrote 1 and 5, you would get 0 points. You must write a complete sentence. Let's take a look at the next one. It says, identify the independent variable if we compare the results from test tube 1 and 3. So now I'm going to look at 1 and 3 here. Okay, so let's note what's different. So it looks like it is identical to 1, except this one is not going to be exposed to the light. Okay, so they're varying the amount of light, or they're changing, or they're wanting to see if light is affecting the rate of photosynthesis. So if test tubes 1 and 3 are compared, the IV is the amount of light. Amount of light. That's what they're changing. That's what they are making different. Okay, so C says identify the negative control in this experiment and suggest information that this test tube And this was supposed to be changed, but I guess it didn't. So let me change this, which says suggest information it will provide. I think your notes say it, not test tube 5. So this kind of gave away the answer. But if it didn't say test tube 5, hopefully you would be able to indicate that out of these test tubes, one of them is the negative control. And in this case, it would be test tube 5. Let's indicate test tube 5. Okay. Now, this is one part of C. I just got 1 point for correctly indicating that the negative control is test tube 5. But then the last point says suggest information that this test tube will provide. Now, let's think about this. If we just put the dye in water and we have a test tube here, no chloroplast, and we shine light, our test tube should not turn clear because there's not going to be any photosynthesis happening. Okay. So going back to what those control groups can tell us, okay, so sometimes they can help demonstrate if the equipment, materials, or the methods collecting data are working as expected. They can also help us identify if outside factors are affecting or not affecting our data. So let's go down here. And I already wrote out the answer. And this is, again, to kind of save time taking notes here. So let's think about this. We know test tube 5 is going to stay blue. What that tells the researcher is that any change in color that does happen in the other test tubes is because of photosynthesis occurring. So it's giving information to the researcher that, in fact, this dye cannot change colors on its own. It will only change to clear if chloroplasts are performing photosynthesis. Okay, so light itself is not a factor that can turn the dye blue. Only the chloroplasts that are doing photosynthesis. So it's proving some information to the researcher in this experiment. All right, let's take a look at this question. And then again, this is an example of where we have multiple independent variables being tested at once in an experiment. So let's get ourselves familiar with it. It says that we want to see how seed germination is being affected by different things. Okay, so it looks like we set up different conditions here. And let's go down here and kind of look at our questions. It says if you compare test tube 1 or beaker 1 and 2. So let's take a look at 1 and 2 here. So 1 and 2. Notice that my temperature is the same. The addition of deoxygenated water is the same, and they did not use any mineral oil to block oxygen diffusion. The only thing that they changed is this right here. So when you compare beakers 1 and 2, then we're trying to see if boiling is affecting the germination of the radish seeds. Let's take a look if we compare 1 and 5. So I'm going to clear this off. Actually, I'll leave it so that you don't get annoyed that I keep erasing writing and then writing over it. So let's take a look at beaker 1 and 5. What is being tested if they just compare the results of beaker 1 and 5? So it looks like everything is the same all through here, but they're changing the temperature. So let's kind of indicate that they're changing temperature. That's going to be our independent variable. So they want to see if temperature is affecting seed germination. All right, it says that we could also have, we can compare the results from 3 and 4. And in this case, it looks like we would be seeing if the addition of deoxygenated water can affect germination. But there are definitely some beakers here that we would never want to compare, okay? And those would be beakers in which there's more than one difference between them. So, for example, let's take a look at beaker 2 and beaker 5. So beaker 2 and beaker 5, you can see that temperature is different. And then also the seeds are boiled versus not boiled. So if our seeds don't germinate, in beaker 2 versus beaker 5, we don't know if it's because beaker 2 was hotter or because it was boiled. Okay, so you can't compare beakers if there's more than 2 changes between those experimental groups. Okay. All right. Moving on to our next page. I wanted to give you a little more practice with that positive and that negative control group. So let's take a second to get familiar with this experiment. It says that We want to see if different concentrations of salt water is affecting the mass of plant tissue. So we measure the mass before, and then we expose them to solutions that have different concentrations of salt. And we look at what happens and determine the percent change in mass of the plant tissue. So did mass increase or did mass decrease? So the question says identify the control group for the experiment as either a negative or a positive control and indicate the information this control provides. So it looks like we have taken plant mass and we put no salt. So basically some drops of water on it. And then this molarity is an indication of concentration. So we put a saltwater solution that has a concentration of salt that is 0.2 moles per liter. And then another salt solution that has a little more salt in it, 0.4 moles per liter. Okay. So we're varying the concentration of the salt. So out of these setups, it looks like we took a measurement at 0, at 2. We had a plant exposed to 0.4 and one that's at 0.6 and 0.8 and 1 moles per liter of salt. Which one of those would be considered our control group? Well, I hope that you were able to point out that the plant tissue in which we just placed water on it, okay, that would be considered our control group. So the plant cells placed in a 0M, so no salt solution, would be our control group. Now, is it a positive or a negative control? Because the question is asking for that. Now, let's think about this. We exposed the plant to just drops of water, and then we measured something, and we saw, something happen. We saw the mass change by almost 20%. So we saw a change as expected. Okay. So since we saw a change happen in that group, then that would be an example of a positive control. Okay. Now, this part is hard, and it's just going to take practice throughout the year in order to be able to write or answer a question like we see here in the second part. Indicate the information the control provides. Okay. So again, I wrote an example down here. I am specific to this experiment. So you don't want to say that the plant that was put in the 0M salt solution is the control, because that's the most normal situation. You're going to get 0 points for that. So what this is really doing is it's giving us data. So it's telling us that if you just expose a plant, that you're going to see almost a 20% change in that salt water. Okay. And so then when we get these other data points, like we see a change of 5% and we see a decrease in about 7% of the plant mass, we can use the data from the control to compare to see how significantly changing the salt is affecting the percent change in mass. Okay. All right, let's take a look at this next example here. Now, I like this question because this was a very unique question because they are asking you on your test to draw. Okay, so note that there can be different things besides just writing out answers in paragraphs or in sentences. So let's familiarize ourselves with the experiment. It says seagrasses are aquatic plants that reproduce sexually. Male seagrass flowers produce sticky pollen that's carried by circulating water to the female flowers, resulting in fertilization. So what I'm kind of picturing here is if these were together and water was circulating, then the pollen would go from the male flower, it would go throughout the water, be carried by the water to the female flower, and it would pollinate it, okay? And you would get offspring. It says a researcher claims that mobile aquatic invertebrates can also transfer pollen from the male to female flowers when there's no circulating water. So to investigate this claim, the researcher set up aquariums to model the possible interactions. So it says use the symbols below. So I'm right here on A. It says use the symbols below and the template to demonstrate the experimental design. So they want you to design an experiment to test the researcher's claim that mobile aquatic invertebrates can pollinate seagrass in the absence of circulating water. Okay, so it says draw the symbols in the aquariums, in both aquariums, and do not use any symbol more than once. So let's take a look at our aquariums down here. Okay, so we want to see if we can get pollination when there's no circulating water. So that's something that we would have to indicate here. So there's no circulating water in either of these. Okay. All right. So first of all, we're going to have to establish that it's the invertebrate that is actually transferring the pollen. Okay. And that this doesn't happen by some other outside factor. Okay. So we need to let me go back a little bit here so I can see the symbols here. So we need to put a male flower in the aquarium, and we need to put a female flower in the aquarium. Okay. Now, we would expect that if there's no circulating water, that we should end up with no pollination. And you don't have to write this, but I'm just kind of showing you here what's going to happen. And this is going to help us figure out our if this is a negative control group. Okay. And then we have an experimental aquarium here where we want to see if this invertebrate is going to cause pollination. So we've got to put the male flower in there. We have to have the female flower. And then we've got to add the invertebrate. Okay. All right. Now, if you think about it, negative control, we should see no change take place. So we're going to allow this setup to sit for a certain period of time. We should see no pollination because the water's not circulating. Okay. So that would be an example. We're going to get no change, no pollination. In 10 days, we're still going to see the male flower and the female flower with no pollination. That would be an example of a negative control. Okay. If we also see no pollination over here, now you don't have to write this. If we get no pollination over here, then that means that the researcher's claim is not accurate. Okay. That the invertebrates can't increase pollination. Now, I wanted to go ahead and talk about this part right here. Identify the dependent variable in the experiment. Remember, the dependent variable has to be some kind of data. So whatever you write down here for the independent variable, you have to be able to get a number for it. So you don't want to say that the dependent variable is pollination because we need to be more specific. So what do you mean by pollination? How can you measure pollination? I need a number for pollination, okay? Well, one thing that we could write down is we could actually count. Once pollination happens, you're going to get offspring. So one thing we could say is the dependent variable could be the number of offspring. Okay, and I'm not going to write this whole thing out, but we could say the dependent variable could be the number of offspring. I'm doing what you're not supposed to do. So we'd actually write a complete sentence here. That's data we could collect. Okay, or we could say the number of pollination events. So let's say 6 female flowers got pollinated, okay? Or the percent of the female plants that got pollinated. So again, whatever we put down has to be a number. All right, moving on. So we're going to be doing a lot of experiments, collecting data and graphing that. And graphing, I think students will feel that it's pretty easy, but they usually make you do some pretty complex graphs. In addition, we're going to go over some stuff about graphing because you should be able to look at a graph or a table and in about 5 seconds identify what is the independent variable and what is the dependent variable, even if you don't know what half the words in that graph or in that table mean. When you look at a graph or when you're making a graph, you want to make sure to note that the DV is always going to be on the y-axis. Now, to help you remember the difference between the y and the x-axis, I like to think of this being the long tail of a y. To help you remember, that's the y, okay? So the bottom part, that's the x. And you can turn the y into a d. So that might help you remember that the dv goes on the y-axis. Now, most of the time, the IV, okay, so you can turn the x-axis into an I, the IV goes on the x. But this is not always the case. If you have a multi-line graph, you need to note that the IV is not on the x. You're going to have to study the key. And that's going to then tell you what they're varying, what that independent variable is. Okay. What I mean by a multi-line graph, let's just kind of draw one right here. So if we see multiple lines that go like this, so multiple lines, then you have a multi-line graph, and those lines are going to have a key to them. So each line is going to represent something, a part of the experiment. And so look at the key for the independent variable. Sometimes you're going to be told when you make a graph to make a line graph or a bar graph. Sometimes, though, it's going to say make an appropriate graph, and you have to determine whether to make a line graph or a bar graph. So how are you going to know when to do what? So here's the easiest way to determine whether to make a line graph or a bar graph. You're going to make a line graph if what you're putting along the x-axis needs to stay in order. I'll give you an example of that here in a second. If what is going on the x-axis can be switched around in any order and the graph is still going to make sense, then you should be making a bar graph. Okay? All right, and then the last thing that I have here before we move down, it says we're going to work on spacing, like how to space out those numbers that are going along the y-axis or the x-axis. And don't assume that if you write down 4 numbers, they're going to be evenly spaced, okay? All right, so let's take a look at this information down here. And we're going to go ahead and we're going to try to make a graph here. So it says using the data, draw the graph to show the effect of temperature change on the rate of transpiration. So we just talked about looking at a table, the IV is going to come first and the DV is going to come second. This is going to be a very simple table to set up. Now, we know that the DV always goes on the x-axis. Okay, so since we know that the DV is transpiration rate, we're going to take this whole label and the units, and we're going to put that there, transpiration rate. And even if you don't know what those units are, make sure you put the units exactly like they have it on the table, okay? Now this is going to be an easy graph. It's not going to be a multi-line graph. The only thing that we have left now is temperature. So obviously temperature is going to have to go down here on our x-axis. So let's go ahead and indicate temperature and degrees Celsius, okay? I'm going to go ahead and zoom out here and see if we can get all the information on here. All right, there we go. Okay, now let's show you some key mistakes here. All right, so let's put temperature, degrees Celsius. Notice I use the units and everything that they had up here in the table. Okay, I'll go ahead and I need to clear my ink here. All right, let's try this. Okay, so we're going to put temperature along our x-axis. And we have 4 temperatures. Let me show you the most common mistake. Students will say, okay, this is 20. So they'll say this line right here is 20. And so they skipped 4 boxes to start. And they'll continue to skip 4 boxes. So they'll put a line here and they'll say this is 23. They'll skip 4 more boxes. Now, don't be writing this. I'm sorry if you are. I'm just showing you what students do wrong. And then 27, and they skip 4 more boxes, and they put 28. So the problem is they've evenly spaced numbers that are not evenly spaced. So between 20 and 23, there's a difference of 3. Okay. But if you look then from 23 to 27, there's a difference of 4. And then from 27 to 28, there's only a difference of 1, but we still skipped 4 over. Okay. So that is a common mistake I see students do. Now, we don't have to always start with 0. If we have a temperature of 20, we can establish that this is 20, all right? 20 degrees Celsius. Now, you don't have to write degrees Celsius just as long as you have it already down here. So temperature, degrees Celsius. So I know that's 20, okay? One thing that would really help, make sure that you assign these numbers, if you put numbers on the X, to lines, okay? So this is my 20. I like to draw a line to, hey, this one is 20. Now, let's skip in a way that makes sense, okay? I will skip on this one. I need to go from like 20 to 28. I want to use up most of my axis on the x-axis, but I don't have to use the whole thing, okay? And I don't really want to count by threes because I'm going to have a really short axis. So, for example, if I, skipped and I put this one's 23, and then I skip three more, and if I put this as 26, and I skip three more, and I put this as 29, notice I'm already done. I've already, got to where I can graph the data point for 28 degrees Celsius, but I didn't use much of my graph. So I'm going to go back here. I'm going to space it out a little bit different. I'm going to do, this makes sense to me. I could do 20 degrees, all right? And I could kind of count by twos. So I'm going to make this, so there's 20, 21. I'll just show you here. So here I'm going to skip 4 boxes and I'm going to put 22. Now, I skipped in a way that it's really easy to figure out if I need to know where 21 degrees is. That's going to be really easy to find. Okay, it's going to be right here in the middle. That would be 21. Okay, so I'm going to skip 4 more. And that's going to, again, I'm going to put a little tick mark there so I know that 24 goes on that line, okay? So notice that 4 boxes means 2. 4 more boxes, I'm going to increase by a value of 2. 4 more boxes, I'm going to be at 26. 4 more boxes, I'm going to be at 28, okay? Now, notice that I do have a number here, 27. I don't have to write 27 on the graph. But when I go to plot the number for 27, I just have to make sure that I do it in the correct area, okay? So I'm going to know that right here should be 27. So when I go to put a data point for my transpiration rate, I'm just going to put it on that line, okay? So then someone reading my graph is going to know at 27 degrees Celsius, my transpiration rate is 5, okay? Also, let's go ahead and, again, we should have transpiration rate. If you don't have it, go ahead and put it in there, transpiration rate, and put your units, just like we have here. Okay. And it looks like the highest number that we have to graph is 5. Okay. So I want to use up most of my y-axis if possible, and I need to count up in a way that it's going to be really easy to plot my data here. Okay. So we don't want to do something like count up like maybe in an odd numbers or anything like that. Make it easy, okay? So I'm going to try, how about every 4 boxes is 1? I think that would be pretty easy. So let's see if that works. Okay, so this would be my 1. And then another 4 boxes is 2. And another 4 boxes is 3. Another 4 boxes would be 4. And it looks like this is going to work just fine, because the highest number that I have to graph is going to be a 5, okay? So whenever I go to graph at 20 degrees Celsius, my transpiration rate is 1.5. That should be really easy to find. So my 1.5 should be right here. My finger can get that data point there. So at 23, it should be easy to find 23. So 23 is right here. Okay, I'm not going to actually put the line there. I'm just showing you where I'm at when I'm looking at my graph here. So that would be 23 in the middle of 22 and 24. So we would go up and we would put a data point right on the 3. And then my 27, we said, is in between the 26 and the 28. I just need to go up that line and make sure I don't get off the line there, because if you put one data point in the wrong spot, they're going to take off that point, even if all your data points are correct. And then it should be easy to find 28. So go up the line for 28 and put 4.5. Okay. Now we're going to connect the dots, right? We're going to draw a straight line from one dot to the next, from this dot to this, from this dot to this. I've seen people draw crazy lines, okay? A couple of things I'll see students do. Sometimes if our data doesn't necessarily start at 0. Notice we didn't start at 0 degrees Celsius. Students sometimes always like to connect back to 0, 0, and that's not the case. If it tells you to graph and put a data point at 0, 0, then you'll connect back to it. But we don't always draw a line back to 0 and 0, okay? Also, This little mistake right here, let me show you what not to do. If you would have drawn this line a little bit past your last data point, you would have lost that point for correctly. You would have lost that point for correctly plotting your data. So do not draw past your first and last data points that's given to you in your table. Okay. All right. I think we talked about the rest of the things that I have there in that little box. So be careful with those do nots. I see those things happen a lot. All right. Going on to this page. And going back and discussing some of the things that I mentioned earlier that we already wrote down on the previous page about our graphs. So when we should make a line versus when we should make a bar. Clear that. And here we go. Okay. So remember, if you're asked to plot data, you know to make it a line graph when the categories on the x-axis must go in order. So if they must stay in order along that x-axis, then you should be making a line graph. So here's what I mean by that, okay? So notice, when I look at this graph right here, and I wrote a 2 and a 4 and a 6 and an 8, would it make any sense to go 2, 6, 4, 8, 10 for the amount of carbon dioxide? It wouldn't. So since what we are putting here on the x-axis must go in order, then we should be making a line graph instead of a bar graph, okay? Same with here. It wouldn't make any sense to label the x-axis 20 and then 10 and then 30 and then 40 and then 60 and 50. That would make no sense to rearrange what we wrote on the x-axis. So again, that's why this data was plotted as a line graph, okay? Now, you should be able to look at a graph and in 2 seconds be able to tell me what is the IV and what is the DV. So remember, on a single line graph, okay, actually on all graphs, the DV is going to be on the Y. Okay, so for that first one, the DV was rate of photosynthesis. For this one, my DV is the photosynthetic rate. Let's go ahead and write that down here because the DV is always going to be on our Y axis. Okay, the IV can be a little bit trickier. Okay, so going back up here, if we have a single line graph, our IV is on the X. Okay, we're actually going to put that for all graphs, but that multi-line graph. Okay, so for all graphs, the IV is going to be on the X unless you have a single line graph like we have up here a multi-line graph like we have up here at the top. Okay, so if you want to figure out what the independent variable is, here's the key. See how I have multi-lines? There's a key to those lines. So it looks like my IV equals color. Okay, so only on a multi-line graph is your IV in the key. All the other times your IV is going to be gotten from looking at what is listed along the x-axis. Okay, so let's go down here to our bar graph. I'm going to get rid of some of this ink that I have on here. All right. Maybe this is being weird, but that's okay. I can erase. I'll use my eraser. Okay, so why did we make a bar graph? Well, we made a bar graph because when we are listing our categories along the x-axis, so we had mosquito, honeybee, housefly, beetle. So notice I could have put those in any order and graphed them, and I would be able to draw conclusions. Like I could have listed the honeybee here first, and then mosquito, and then graphed its tall bar there, okay? So it's more appropriate to graph our data as a bar graph if the categories on the x-axis can be switched. If you can switch them around and it still makes sense, you can still draw conclusions, you should be making a bar graph and not connecting dots in a line graph. Okay, so this one right here, the double bar graph. So I could have put nitrogen first and graphed it and then graphed the phosphorus over here. Or I could have graphed this one first. I could put these in any order, graph the data, and still draw conclusions. So that's why it's more appropriate to make this information in a bar graph, okay? Again, remember that in a single bar graph, the IV is always going to be on the axis or on the X. In a double bar graph, also on the X. The only difference where we're going to have the IV that is not on the X is when you have a multi-line graph like you see here. You got to take a look at the key. All right, zooming on. This is just showing you that sometimes people draw crazy lines and you can lose a very easy point. So notice this person right here, they have very kind of smooth, curvy lines. So they're actually indicating data that they did not get. That's not true. We need to make sure we connect straight from one data point to the next. Here's just some other crazy lines I've seen people do, and this one, a really wavy one. Also, notice they went past that first data point and the last data point. So make sure you don't do that. All right. Let's move on here to this next example. I'm going to zoom out a little bit and I'm going to clear my screen there. Okay. Now, after going through where to put the IV and the DV, I want to see if you can set up this graph for this experiment. And this is a pretty complex graph. I think this graph caused a lot of issues for students whenever it was on the AP exam a few years ago. Okay, so on this one, it says on A, on the axis provided, construct an appropriate labeled graph to illustrate the effect of caffeine on the probability of bees revisiting a nectar source. Okay, all right, the easiest way to start is look at the data that's being collected. So these are the numbers right here that are being collected. And it tells you up here that those numbers mean this. This is the probability of revisiting a nectar source. And then they give you some units here, which you're going to learn about that here in a little bit in lecture 2, actually. So we have right here, we have our dependent variable. So we're going to write this as is. This is going to go on that y-axis, okay? All right, I want you to pause because now here's the hard part. Try to figure out what you're going to put on your X. Are you going to put control of caffeine, or are you going to put the 10 minutes and the 24 hours? Because those are kind of the two pieces of information that are remaining that have to be indicated somehow on this graph. So take a second to decide. All right. So remember, anytime you have a bar graph, you should be able to put the independent variable on the x-axis, and you could put them in any order and still draw conclusions. Now, notice that there's only 2 times. We have 10 minutes, we have 24 hours. They're not even in the same unit. So you're not going to be putting time along the x-axis, okay? So it looks like we have a control. We have no caffeine versus when we do expose those bees to caffeine. That's my independent variable. And the independent variable is going to go on the x-axis here. So we would have the control, okay, versus caffeine. We're just going to make a bar graph here. Oh, I spelled that wrong. E-I-N-E for caffeine. I know that looks bad. My other lectures, I promise, my writing is going to be a lot better in those. Okay, so now what we have left is we have to indicate for my control group, okay, we're going to have 2 data points. We have 10 minutes and then we have 24 hours, okay? Now, I'm going to go ahead and put some numbers here on the x-axis because these are decimals. I think decimals can sometimes make it difficult for you guys to graph, but honestly, kind of just, you can kind of treat them like whole numbers. So instead of 0.83 as something we have to graph, think of it as being like 83, okay? So let's go back here. It looks like the highest number that we'd have to graph is around 0.83, okay? So let's start figuring out how we're going to divide up our axis here. And I'm thinking that we're probably going to have to count up by 0.1. So 2 boxes can mean 0.1. Okay, so this is like 0.10. Okay, if we go up 2 more boxes, 0.20. So this is like 20. Now we can count up 10, 20, 30, 40, right? This is just, we have a decimal in the front, okay? So go up 2 more and put my line here. This is going to be 0.30, okay? Go up 2 more, 0.40. Okay, so don't let decimals be too scary. If you kind of just eyeball them and make them kind of like their whole numbers. I know they're not, we're not just counting up by 10, 20, but we are just going up by 10 basically. And then finally we have 0.80, okay? All right, so for the control, it looks like we have 2 bars here. I need to go back up a little bit. So 0.72 is 10 minutes, right? You have to graph your data completely accurate to get a point. Each of these 4 bars. If 1 bar is off, you're going to get 0 points, okay? So I'm going to make a bar here, and I need to go up to 0.72. I'm doing the best I can with my finger here. Okay, here we go. All right. And then the other control, it looks like we have is 0.41. So let's go up. Let's do another bar. We can put it right next to it, or we can skip over. It doesn't matter. But we have to make sure that we are exactly at 0.41. So I got to find that bar. Looks like it's right here. And I got to be a little bit above it, 0.41. Okay. All right. Now we are going to have to make a key in this situation because I need to know which one goes with the 10-minute mark and which one goes with 24 hours, like you can see up here. Okay. We can do it 2 different ways. You could actually just label this bar. I mean, you can put 10 minutes down here. So underneath this bar, you would put 10 minutes. Okay. And then underneath this other bar, you would write 24 hours. And I apologize that I'm zooming out so that you guys can see more of my graph, but I can't write that small. So we can also put 24 hours. I'm going to change it to blue. So right here, we can put 24 hours to go with that bar, okay? We also could have made a key if we wanted just to put dashed lines on this one, and then we can make a key over here that says that whenever we have a bar that is dashed, okay, so here's our dash bar. This means 10 minutes, okay? And then the one that is not dashed here, right, that one was 24 hours, okay? And then we would go ahead, I'm not going to do it just to save time on this video, but we would also then have 2 bars for the caffeine one, okay? All right, let's move on here. All right, sometimes you might be asked to design an experiment. And remember, we just talked about IV, DV, constants of the control group. So if you're asked to design an experiment, then these are the things that a grader is going to be looking for. So 1, have you established an independent variable? Do you have a difference between 2 setups or 3 setups or 4 setups? So you need to tell me what is changing, and you need to be very specific. So just don't say vary temperature. Tell me to use 10 degrees Celsius and 20 degrees Celsius. Don't say change the salt concentrations. You need to say change the salt concentrations and tell me. Use a 0.1 mole per liter solution, a 0.2 moles per liter solution, etc. You need to tell me what is the dependent variable. Remember, it has to be measurable. It needs to be something that is telling me a number to collect. So write down the number of species or record the temperature of the water or etc. So it has to be a number. You need to mention at least 3 things to keep constant to get that point. You need to identify a control group if you can, because sometimes some experiments, it's hard to establish a control group, but if you can, that would give you a point. If you tell me to do things like take many data points and then maybe calculate an average, or if you indicate to use a large sample size, or if you say repeat to verify the results, then that'll give you this point for verification and replication. And then finally, if you write out your hypothesis or your prediction, then that would give you another point. All right, so let's go down here to, here's an example. I'm going to go right down here to C. So this question says, design a controlled experiment to determine how the diversity of plant species, so how many different types of plants, in a newly abandoned field would be affected by large herbivores, okay? So again, to get points for a experiment, for designing one, these are the things that you would have to mention. Okay, so I just kind of list them here. You got to tell me what to change, what to measure, variables to be held constant, identify a control setup. So that would be maybe a field where there was no herbivores to compare to a field and the number of plant species when we are using herbivores. So I'd have to say make sure we use a large field or a large sample size, repeat, etc. And I'd have to give a hypothesis. On the next page, I have an example of something that a student wrote. So this is a real answer from a real student that was released. Okay. And let's take a look at their answer and see how many points. Now, when you go to write this, there's usually a cap on your points. For example, I said that each one of those things would give you a point. So technically, that would be 6 points, right? But they usually cap it at about 4. Okay. So let's see how many points they got out of the maximum, which would be 4. So in this one, I know it's a little difficult to read, but it says to create such an experiment, one needs 2 separate areas, both of the same type of flora. So here's a constant, except one will contain herbivores and the other will be allowed to grow on its own. Okay, so they set up something different. One pasture has herbivores and the other one doesn't. So technically, they get 1 point for identifying a change or identifying the independent variable. It says just let the two sit for some time, several years. Okay, so this actually is telling me to let this go on for a while. Okay, so that's means that we're gonna get a lot of data since we're doing this for years. Now it says compare the type of diversification. I have no idea what that means. What am I supposed to count? So at the end of 3 years, I need to go and write down a number for the pasture with herbivores, and then a number for the pasture without herbivores. But they don't tell me what I'm supposed to count and write down. Okay. And then they did say one must also ensure that both regions have equal access to pollinators, the same amount of sunlight, and the same amount of water. Okay. So they did give me 3 constants. So I give them 1 point for the independent variable. Okay. I would give them 1 point for telling me those 3 things to keep constant. And then I would actually give them a point for doing this over a long period of time, and that's going to increase the validity. So I would give that person 3 out of 4 points for that design. All right. Now, this one, I just wanted to point out, I'm not going to go through all of it, but it says design an experiment. Design experiment using artificial flowers to investigate potential negative effects of increasing caffeine or concentrations in nectar on the number of floral visits by the bees. So the number of visits by bees would be the dependent variable. Okay. It says specifically after that, identify the null, give me a control that you would set up, and predict. So in this case, the only way you're going to get 3 points, and all you're going to do is get 3 points on this section, is to do those things. You don't have to list the IV, you don't have to list the DV. You're not going to get any points if you list those things because they're looking for you to do these exact 3 things right here. So you think of an experiment, and then you would state, this would be my null, this would be my control, and these are the results that I would predict. All right. And I think we're actually going to stop there. I'm not going to worry about this page. I don't even know if I'm going to print it out for you all. So it might not even be there. And that concludes your very first lecture with a few more to come eventually after this.